Welcome back for the second part of our carbohydrates lesson. In this particular video, we're going to look at the structural and functional diversity of polysaccharides. This is a slide from that last video, and I just wanted to highlight that we looked at these bonds formed between the anomeric, remember that one that had been the carbonyl carbon of glucose and a hydroxyl group of another molecule. And we talked about the fact we could have O glycosidic bonds, so we have that oxygen in here. Or we can also see that same type of bond occur with a nitrogen, okay, as that connector. So what we're going to do first is look at some derivatives of carbohydrates. Before this stresses you out, you do not need to memorize all of these. I just really want to highlight that there is a large diversity of carbohydrates. It largely comes from the ability of all of those hydroxyl groups to be substituted out for something else. So when it's a derivative, it has the base of a carbohydrate, but it's no longer just CH2O, right? We've added some other things to it, so it's no longer just that. If we look at some examples here, just to look at some options, okay, this D-gluconic acid has a straight chain version on the left and a ring version on the right. Okay, so straight chain, ring version. And this is where glucose has been oxidized. So now, instead of the aldehyde group, we have this carboxylic acid group or carboxylate group. And here's the straight chain version, even though we're showing it wrapped around. And we can form this bond. And notice, so this is called a lactone. Okay, when this ring closes up on a carboxylate group rather than an aldehyde or a ketone. And so notice that right here we have this carbonyl instead of hydroxide. All right, let's look at another derivative of carbohydrates is sorbitol. You've probably heard of this. It's found in fruits. It's an additive to medicine and foods because it has about a third of the calories of sugar, so it's a lower calorie sweetener. Your body has the ability to metabolize it, so it can be converted to fructose and then to glucose if needed, but it can also be built up from a dysfunctional glucose system. So for example, if you have too much sugar in your blood from diabetes or something, we can start building up some of this sorbitol. So when we look at this as compared to sugar, we're going to call this a sugar alcohol. And we see that we have that OH group instead of the aldehyde, right? So the carbonyl was reduced from an aldehyde to an alcohol. So that would be our sugar alcohol. We can also have amine sugars. So here we see these amino groups substituted in for some of those hydroxyl groups. Okay, these are two different, glucosamine and galactosamine. These are important starting materials for some polysaccharides we will discuss in a minute. Okay, more derivatives. So notice we had the amine group, and now we're adding this acetyl group to it. Okay, so these are other important monomers for some polysaccharides. And in this case on the right, this one's just interesting because it's got a bunch of carbons. It's not really normal. So this sometimes serves at the end of long polysaccharides attached to a protein, sometimes does some cell-to-cell signaling. Mostly interesting just because it's bigger than most of them. All right, another one that we'll be returning to look at is this fucose, if I can talk. When we look at this one, we'll look at this process, we have an L version, and that's really the thing we want to highlight here. We said that normally the D version is the biologically active, but in this case, the biologically active one is the L version. And what we end up seeing is that this is a common piece that gets added on to oligosaccharides or polysaccharides to give functionality to those. And it's likely because there are some enzymes that recognize this L version. So that helps with their specificity. Anyway, you get the idea. All right, we're still going. More derivatives of carbohydrates. This muramic acid or acetylmuramic acid is a just highly substituted monosaccharide. Like there's lots of stuff on here added on. And they're important components of bacterial and plant cell walls. All right, so if we go back to that original picture, really what I want you to get the idea of here is that carbohydrates provide so much more diversity. You know, usually we just think of simple sugars, we think of glucose, we think of glycogen, but because of all those hydroxyl groups that can be easily substituted with other things, we end up with numerous different molecules that are biologically important. So we're going to look at a few of those. Again, you don't need to memorize any of these specific ones we just looked at. But what we do want to look at are saccharides that are bound to proteins. All right, so as we talk about saccharides that are bound to proteins, it works in both directions. The saccharides often bring more functionality to the protein, and the proteins bring more functionality to the saccharides. So it's kind of a win-win situation. So we're linking together two different classes of biomolecules, and they get classified into three broad categories. So we have the glycoproteins, the mucins, and the proteoglycans. If we start over on the left, the glycoproteins are predominantly protein, but have oligosaccharides covalently attached through N or O glycosidic bonds. So the glycoproteins are mostly protein with a little bit of the saccharide. If we go over to the other side, the proteoglycans, these are polysaccharides with a small protein core covalently linked by those same N or O glycosidic linkages. So now they're mostly polysaccharides. Notice both of them use these glycosidic linkages, those ones we looked at in that very first slide again. Proteoglycans are maybe about 5% protein. They tend to be show up in connective tissue, shells. These glycoproteins tend to be antibodies, membrane proteins with oligosaccharides bound to them for some sort of cell-to-cell signaling. They're active in immune response. And then right in the middle, we have these mucins. So these are between the two extremes. They're about half protein, half polysaccharide. Glucoprotein, this is a glucoprotein that's a part of mucus. All right, let's look at these three classes a little separately. So glycoproteins, we said these were the ones, let's go back up here, were mostly protein, right here on the left. Okay. And in all of these, again, this is the same picture we looked at before. We are going to see that protein bound to the oligosaccharide with either an N linkage or the O linkage. Okay? And remember that the N linkage generally comes from asparagine, or the O linkage is either serine or threonine, those ones that have those hydroxyl groups on the side chain. So as we talk about the glycoprotein, all N-linked polysaccharides consist of a common pentasaccharide core. Think about that, pentasaccharide, 5 sugar core that consists of 3 mannose. So that was on one of our slides from last time. Not something you need to memorize, but it's a 6-member sugar. And then 2N-acetylglucosamine. So those were on our ones we just looked at a minute ago, those derivatives. So think glucosamine. You can see that amine that was substituted. Okay, and then we have that carbonyl. And then, along with this, there's another 6-carbon sugar. And it can have more monosaccharides. So the part highlighted in gray here is that pentasaccharide core. And you can see it essentially always looks the same. We have the acetylglucosamine units, and you'll see that we have a kind of standard abbreviation. You don't need to memorize it. And then we have the mannose, also abbreviated. And then there'll be other pieces. Okay, so some kind of things that are things to note. We will often note the linkages between these different groups because those linkages are important, right? Those linkages determine the three-dimensional structure, and structure tells us the function. These monomer units, besides having like the shorthand of MAN for mannose, they also have these shorthand symbols. And you don't need to memorize these, but this blue square will always be N-acetylglucosamine. This green circle will always be mannose. And so it's an easy way for us to draw and represent some of these things in a shorthand. So let's look at an example of a glycoprotein. So protein glycosylation patterns for blood groups. So if we think about this, human ABO blood group reflects specificity for why can't I talk today? Glycosyltransferases. So when we look at these, right? When we look at these transferases, enzymes, they transfer groups, and in this case, they're going to transfer sugar groups. So transferases transfer groups, and now we're going to transfer these monosaccharides or oligosaccharides to a protein or a lipid. Okay, so we're transferring these saccharides to a protein or a lipid. So if you have a gene for a specific one of these transferases, then you'll be able to build a specific type of the oligosaccharide. So all blood groups share the oligosaccharide foundation of O. So they all share this piece. All right, so it's galactose, N-acetylglucosamine, another galactose, and then a fucose. And we can see that we've got our shorthand here. No, you don't have to memorize that. But if you have an A blood type, you have a specific enzyme that will add, let me highlight in a different color, an N-acetylgalactosamine, okay, right here to that galactose, right here, okay? And if you have a B subtype, you would add galactose right there, okay? If you have AB, you're going to have the enzymes to add both of those. And then the blood type O has none of these active enzymes. So if you're transfused with a blood type that's not your own, your body's going to see it as foreign and attack it. And the thought about why there are these different blood types in humans has to do with viruses. And this is just a theory, but viruses have evolved to bind to certain oligosaccharides on surfaces of cells. That's the route that viruses can get into cells. So the thought is that within a population, if you have different people with different blood antigens, even if you end up with a virus that bonds to a specific antigen, it wouldn't wipe out the entire population. So for example, if we had a sudden pandemic of a virus that bonded to this galactose piece, well, people with the B antigen are in trouble, but everybody else is okay. One theory. about why there are different blood types. All right, let's talk about proteoglycans. So proteoglycans are proteins attached to glycosaminoglycans, which make up 95% of it by weight. So if we go back up here, dun, 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 the glycoproteins, right, were mostly protein, but the proteoglycans are mostly polysaccharide. Okay, and by mostly, we mean about 95% by weight. So these glycosaminoglycans, big word, that are composed of repeating units of a disaccharide. And it's a derivative of an amino sugar, so we can expect to see an amino group. Let's look for those. Right there, right there. Where is our amino? There we go, right there. Amino sugar, and which carries a negative charge, either as carboxylate or sulfate. So we can see up here, we've got this negative charge. Here we go. Up, we got a bunch over here. So these proteoglycans are key components of extracellular matrix and serve as lubricants. So when we look, a few other things I wanted to point out. What color haven't I used? If you see these squiggly lines right here and here, what this is saying is that this is the disaccharide, but now we're going to have repeating units of this going off in each direction. And here we're going to see those there and there. They're not quite squiggly lines. As we look, the hyaluronate is often a component of cartilage. Heparin is something that's released at the site of injury. These negative charges are important because those negative charges will attract water molecules. And so it allows for the idea of these hydration spheres around the molecules. So that essentially these polysaccharides carry lots of water with them. And that water is going to give some of the characteristics and functionality. And we'll look at that in our next example. So here's an example of one proteoglycan. Let's look at the components of it. So when I look at this, I have a backbone of this hyaluronic, I cannot speak today. Let's go back up here. This one, hyaluronic. Anyway, we have our backbone. You get the idea. Right here, it's this long blue thing. And if we go back up and look at this, it has a bunch of negative charge. So there's all this negative charge right along here. Negative charge all along here, along this backbone. All right. Then, when we look at this purple that I've shown in purple here, this protein part. So this whole thing is this kind of protein. But we said this is mostly polysaccharide, and that's because in this long protein, right here is incorporated some of these glucosaminoglycans. And these are these really big polysaccharides. And again, they have sulfates on these particular ones. And so there's lots of negative charge around these things too. Okay. And these units, this unit right here is repeated over and over and over. These circle parts are globular regions of the protein. And then we'd have this glycosidic linkage right here to hold that protein to the backbone. If we come over to the left, we have an electron micrograph of this protein. And so you can kind of see that backbone. You can see all these pieces sticking out of here. All right. And this one actually works as a shock absorber. So remember all that negative charge, and so there's all these water molecules around it. You can picture just tons and tons and tons of water. So we've got this whole hydration sphere, right? And if you think about, say, you go out and you jump off a wall, land, and as you land, it creates pressure on this whole area, and that pressure essentially squeezes out the water. So the water gets pushed out. Whoops, there we go. So the water gets pushed out. And in doing so, it absorbs some of that shock. It has protected you from some of that shock. And then as you relax and that pressure releases, the water comes back in, rehydrates. Okay, so that ability of the glycans to be hydrated with water is key for the functionality in this point. All right, let's look at mucins. So as we talk about mucins, they are high molecular weight proteins. So this top part is the protein part. It's a picture of the protein part. They have long stretches of serine and threonine amino acids. So VNTR segment right here stands for variable number of tandem repeats. Essentially, all through here are a bunch of serine and threonine amino acids. And so those serine and threonine amino acids can make those O-glycosidic linkages. So then if we want to look at the saccharide part, down here, this is our polysaccharide part. Notice over here, we have our serine. That's not what I wanted. I wanted the highlighter. We have our serine or threonine. So those serine and threonine are all along here. Okay. And then we can look at our shorthand, okay, for our polysaccharide. This would be different for different ones. I've got the little key over here. You don't need to memorize any of this, but notice, for example, the yellow circles are galactose. The yellow squares are N-acetylgalactosamine. So there is some logic to it. Blue squares, N-acetylglucosamine. So we have this polysaccharide part. And up here, each one of these serine or threonine can bond to one of these whole things. So you've got these hanging off the sides up there. Okay, this is about 50% polysaccharide, 50% neutral saccharide. I'm sorry, negatively charged saccharides, and 50% neutral saccharides. So they will hold water, but not as much as the proteoglycans we were looking at before. The saccharide composition varies from cell to cell, organism to organism. Let's see, what else was I going to tell you about this? Oh, these kind of end areas, the D domain and cysteine-rich areas, are areas that help these proteins polymerize, so they kind of clump together and make the mucus more viscous. So mucins are synthesized in specialized cells, for example, in the gastrointestinal tract, to help protect the lining. All right. So we have looked at a lot of detail, but the big picture is I just don't want you leaving the class thinking that saccharides are just glycogen and starch and nothing else. So you don't need to memorize most of these structures or anything, but what you should know is how these carbohydrates and proteins are attached to each other. So those O and N-glycosidic linkages, which amino acids are used for those. And you should be able to differentiate glycoproteins, proteoglycans, and mucins, okay, based on the structure and function of those biomolecules. And that is mainly it. All right, that is it for this video.